denature a protein, what happens? Definitely, you are going to have when you denature, definitely you are going to lose the function. Okay, a protein will have its function if the protein structure is intact. If the protein structure is deranged or removed, or you are going to bring it from the highest level to the lowest level or the basic level of organization, definitely you will lose the function of the enzyme. Now, when we tell function of the enzyme, what is enzyme doing? Enzyme is converting substrate into products. So enzyme converts substrate into product. How they are going to do? Is there just a magic where the presence of enzyme can convert into a product, substrate to product, or is it specific mechanism by which this is happening? Definitely yes. We have learned that there is something called as substrates. Substrates is nothing but on which enzyme will act. Okay, it is the one on which enzyme will act, and once it is acting, the substrate is converted into a product. Okay, substrate is converted into a product. So that is about the enzyme. And next, when we see about the product is one which is formed from the substrate. And normally this can happen otherwise also. Okay, the substrate to product conversion can happen in absence of the enzyme also. But what is the enzyme doing then? What is the enzyme doing is enzyme will convert the substrate into product at very faster rate. Okay, at a very faster rate. That's why the enzymes play important role. If substrate to product conversion in the absence of enzyme, we imagine it might take 1 hour. Same conversion of substrate into product in the presence of enzyme will take only 1 second. So that is what the difference makes the enzymes. So enzymes will convert the substrate into product at very faster rate. When we see the characteristics of the enzymes, okay, when we see characteristics of the enzyme, what they are made up of, they are most commonly catalytic action. Okay, they are called as catalyst, and most of them are protein in nature. What they do to the substrate is they lower the activation energy. Last time I told you, if for example, if this is the mountain you have to climb and throw the ball from here. Okay, you have to move the ball from here, which is substrate, to a product. What is enzyme doing? Enzyme is cutting down this edge. Now what happens? It has lowered down the energy required to transport this substrate to product. So activation energy, the energy required for the activation of the process where substrate can be converted into a product is lowered. Activation energy is lowered. Next, because the activation energy is lowered, what happens to the reaction rate? Yes, the reaction rate increases, and the enzymes are heat labile. Because enzymes most commonly made up of proteins get denatured when we give the heat. So definitely the proteins, the enzymes also will be heat labile. They are not much heat stable. Most of the biological enzymes which are present in our body. There are enzymes which are stable at very high temperature also, but normally what we have the enzymes or the human body enzymes are heat labile, and they can be precipitated like any other protein. Okay, they can be precipitated like any other protein. Normally, enzymes also contain something called as cofactors or coenzymes. So the factors which are helping to increase the rate of reaction means they are stabilizing the enzymes itself. So they help in the enzyme reaction. Most commonly, the cofactors can be either organic or inorganic compounds. If it is organic, most commonly the water soluble vitamins. Okay, their active form are most commonly seen along with the enzymes. Inorganic, it can be any mineral, like for example, zinc or iron, okay, or copper, manganese, magnesium. So all these can be the metal ions which are associated with the enzymes as cofactors. What will these do? They will stabilize the enzymes. They can work in absence of these cofactors also, but the rate of reaction will be slow, and also the stability of the enzyme will be low. So that's why these cofactors help. They are like friends. They are doing one function. They are converting substrate to product. Sometimes enzyme cannot do alone. They need some other friend to accompany them. So these cofactors will act as a friend who will come along with the enzymes and convert the substrate into a product. Okay, a substrate into a product. Enzyme will be there. Plus, along with that, a cofactor will be there. Cofactor also will be there for the reaction. So this is about the very basic of the enzyme what we have seen in the previous sessions. Now, when we go ahead, we learn that substrate is converted into product by enzyme. Now, someone can ask you, just merely presence of enzyme can convert the substrate into product? No. What should happen? It should act. The enzyme should act on it. How the enzyme acts? The enzyme acts on substrate. The enzyme acts on substrate with specific site. If you see the enzyme, if this is the enzyme and this is the substrate, okay, this is the substrate. So there is specific site on which it will bind or it will attach with the enzyme. So that is called as what? That is called as active site. So just merely presence of enzyme will not convert substrate to product. There should be presence of the binding between the both. There should be some interaction between the both. So that interaction is not anywhere on the surface. It is very specific site. What site? That is called as active site. So active site is nothing but small cleft, okay, small cleft over the surface of the enzyme where substrate can bind and convert it into a product. So that is called as active site. Now, if someone asks you, what is an active site? It is just a small cleft or small pouch like or small what you call the hole like structure where the substrate can bind. And what are the characteristics of this? It is a pocket like structure. Active site is like pocket like structure. Substrate will bind to the active site only, which is also called as catalytic site. Now, is it specific? Yes, it is specific to its substrate. What is binding to it is always specific. And is the active site flexible or is it rigid? It is not rigid. It is little flexible. What are the amino acids most commonly seen here? It is the hydroxy containing. The most commonly is the serine. Other than serine, you can also see aspartic acid like glutamic acid or cysteine, etc. can also be present. Lysine, histidine, all this can be present. Now, substrate binds with the enzyme. Now, is it permanent or it is just temporary? Okay, this binding is non-covalent, that it is not permanent, and also it is weak. Okay, it is weak. And because this is non-covalent and weak, is it reversible or irreversible? So definitely it is a reversible. When substrate can bind to enzyme, it can have two things. Either enzyme is separated with the substrate or enzyme is converting the substrate into a product. So it can happen two ways. So it is a reversible binding. So this is about the active site and the enzyme brief introduction. Okay, brief introduction. So we'll go to the today's topic. What are the regulations? How the enzymes are regulated. Now, when we see anything in the system of the body, it has got regulation. Like for example, you take the diet, you eat food. Okay, when you eat the food, to digest the food, okay, to digestion of the food, definitely we require what? We require enzymes. We require the enzymes. And the concentration of this enzyme is dependent on the availability of the food. Okay, it is dependent on the availability of the food. If there is no food, if there is no substrate only, do you think enzymes are required in high concentration? No, we don't require the enzymes in the high concentration because there is no substrate to act on. When you take the food, like you have taken carbohydrate diet, don't you think that the carbohydrate digesting enzyme concentration should increase in the GI tract so that the food can be completely digested and the digested food can be easily absorbed? So in general terms, definitely yes, we do understand that there should be some regulation of the enzymes depending on its environment. What do you understand by environment is depending on its substrate. Is the substrate present? Yes, then the enzyme concentration should be high. The function of the enzyme is required at that moment of time, then definitely enzyme should be working. Sometimes enzyme is present, okay, enzyme is there, but it is inactive. Do you think it is serving the purpose? No, we have to convert this inactive form into a active enzyme so that it can function on the substrate. So that is how the body regulates. The regulation is very important. Whenever the enzyme concentration is required, it should increase the enzyme concentration, which will increase the rate of reaction, and it will serve the purpose. Whenever enzyme concentration decreases, it will lead to decrease in reaction rate, and definitely it will also serve the purpose because we wanted to stop the reaction and we wanted to decrease the reaction. So definitely it will again serve the purpose. So enzyme concentration can go increased or they can go decrease. We can increase or decrease, and this part is very crucial. Okay, the regulation of enzyme is very crucial for metabolic control of all the metabolisms. Okay, like for example, if you are converting glucose to pyruvate, definitely there is some regulation. Okay, you have to regulate how much is controlled under that and how much should direct it towards the glycogen synthesis. So that's how the body will do all these manipulations or the changes which are required to divert the pathways. It can divert the pathways or it can regulate the pathways, regulate the whole reaction pathway. Now we all know most of the metabolisms. Okay, most of the metabolisms are not single reaction. Okay, they are not single reaction. They are the sequence or series of reaction. Okay, they are series of reactions and they contain multiple enzymes. They contain multiple enzymes. Now you can ask me, sir, is all enzymes are regulated or only few enzymes are regulated in the series or in the whole pathway? Answer should be only few. Only few enzymes are regulated. Not all the enzymes need to be regulated. The few which will influence the highest on the pathway, they are called as key enzymes or key regulatory enzymes. Means what? They are the enzymes. If you regulate them, then they will impact the whole pathway itself. It will impact the whole pathway, the direction of the pathway, and the rate at which the pathway is going towards its end result or end product. So that is the key enzyme. And on the reaction, like substrate to product, what reaction it is doing or it is present, that is called as rate limiting step. Okay, that is called as rate limiting step, because if the enzyme here stops, the whole pathway stops. Okay, the rate limiting step is a very key step in the metabolic pathway, which will define or direct the rate of reaction, and also it will define the direction of the reaction. Is it A is converted to B, B to C, or is it C to B? Okay, the direction of the reaction is decided by this rate limiting enzymes. So this is about the just a brief of how regulation and why regulation happens. That is the significance of the regulation. Now, when we see the types of regulation, enzyme regulation, okay, enzyme regulation can broadly classified under short-term regulation and long-term regulation. Okay, either it can be a short-term regulation or it can be a long-term regulation. Short-term means, like for example, at the point of digestion, say about. And these short-term regulations are very effective and very fast. Okay, you want to stop the enzyme, you just switch it off. You want to start the enzyme, you just switch it on. Enzyme is already present. Okay, there is no change in concentration of the enzyme, most commonly, or no new synthesis of the enzymes. The enzymes are already synthesized and they are present either in stored or the functional.
Class.
Long term is the one where there is change in the concentration of the enzyme and it is regulated at the level of the DNA or protein synthesis. Okay, it is regulated at the DNA process, that is the protein synthesis process. Okay, short term, again, further it can be of two types. One is covalent modification and other is non-covalent modification in the enzyme. Okay, non-covalent are also termed as allosteric. Okay, also allosteric regulation or modification comes under this, that is the non-covalent. Long term, you can have two things. One is hormonal control, other is the gene regulation. Gene regulation means you are going to either synthesize the protein, that is a translation process. Hormones, again, they act as the messenger where it will tell the cell that it has to increase the synthesis of the hormones or, sorry, enzymes, or decrease the concentration of the enzyme. So again, they are for long term, for a longer time, and they will take some time to set in also. They are not immediate. They take some time to set in, and they take time to change the concentration, and whatever changes are there, that will stay for a little longer time. So this is the enzyme regulation. There is short-term regulation, long-term regulation. This is the important 3-mark or short answer question in the exam. Short-term regulation, long-term regulation. Short-term, two types: covalent modification and non-covalent modification, which can also be a allosteric modifications or allosteric regulation. Long-term, two types: hormonal regulation and gene regulation. So these are the classification of regulation. Now, in the exam, they'll ask you what are the types of regulation. Okay, types of regulation, and describe each, which can be a 15-mark question in the exam. Okay, regulation of enzyme is an important question. So what are the types of regulation? Or sometimes they'll ask you 3 mark. Just write different types of regulation of enzymes. First one, we term it allosteric. Okay, allosteric regulation. Okay, allosteric regulation. Second, we have covalent. Okay, covalent modification. Covalent modification. Third one is called as feedback regulation. Okay, feedback regulation is the third type. Then we also have the other types which act on the gene long term. That is called as induction, that is inducing something. Fifth one, repression, means repressing or decreasing the concentration or the suppressing the synthesis. Last one is compartmentalization. Okay, compartmentalization, which is the enzymes are present in different compartments inside the cell. Now, for example, what are the compartments, you might ask? Definitely, yes. The cytoplasm is one compartment. Okay, mitochondria is one compartment. Nucleus is another compartment. Golgi apparatus, one compartment. Lysosomes, peroxisomes, okay, or the vesicles. So all these can become one compartment. They are separated by a membrane. They have their own matrix. So they are separated from each other. The compartmentalization of the different enzymes itself can be a regulation. Okay, so these are the 6 types of regulation. So in exam, they'll ask you to write in a describe or detail, which can be a 15-mark question, or sometimes they'll ask you to write different types for under 3-mark question. Okay, they'll ask you to write under 3-mark question. In this, especially, the allosteric regulation can be itself a 5-mark question, short essay, and also the compartmentalization can be a 3-mark question, that is short answer. So this is about the types of regulation. So types of regulation. So we'll see one by one what are these types of regulation. Okay. Now first we'll see allosteric regulation. Okay, allosteric regulation, as I told you, it is for 5-mark question. Now, very basic, we all know that enzymes are what? Enzymes are biological compound which are protein in nature, and they help in catalytic reactions. So they are also called as biocatalyst. They have got one site called as active site, at which the substrate will bind. Till this, it is very good. We'll understand. But it was found that when some molecules are introduced into the system, like this is a solution, when some molecules are introduced into the system, the reaction rate either increased or it decreased. The rate of reaction either increased or decreased. They could not identify why this is happening so. Then later they found out that what this molecule was doing, which was added inside the solution, was it was binding to the enzyme. It was binding to the enzyme other than the active site. This is the active site where the substrate will bind. Other than the active site, these molecules were binding, and it was actually either increasing the rate of reaction or decreasing the rate of reaction. So they were altering the enzyme activity by binding on other side. Like they are regulating them. Either they are increasing the rate of reaction, like you using the fan during your summers. Okay, you can switch on and switch off. There is one button, either you can switch on the enzyme or switch off the enzyme, but also you can regulate the enzyme. You can regulate the rate of reaction. So you can imagine this is the regulatory place where binding of a molecule will either increase the rate of reaction. If binding of this what you call the molecule to the enzyme increase the rate of reaction, it is called as activator. Okay, because it is increasing the rate of reaction and activating, it is the activator. The one which is decreasing the rate of reaction, we term them as the inhibitor. It is inhibiting the reaction. Okay, it is inhibiting the reaction. So what is the allosteric site? Allosteric site is the site other than the active site. It is not same. It is other than the active site, which is present on the enzyme itself. Okay, it is present on the enzyme itself. To which, who will bind? Is it substrate? No. To which a molecule will bind, either it is a inhibitor, okay, either it will inhibit or it is a activator. Okay, either it is activator or inhibitor. They both can bind to the site which is other than the active site. This is the active site. This is the allosteric site. Okay, so allosteric site, the allosteric inhibitor or activator will bind. So that is called as allosteric regulatory enzyme or regulatory molecule. Okay, allosteric regulators. What they will do, either they increase the reaction or decrease the reaction. If it increases, it's an activator. If it decreases, it is a inhibitor. So that is about the allosteric regulation. Allosteric regulation. What normally they do when, like for example, we'll take one instance. We have the substrate. It is of this shape. Definitely enzyme, also active site will be complementary, or it will make the changes in the active site, because we told active site is flexible. So substrate can modify the structure. This is the place where the modifier will bind. Now when the inhibitor binds, okay, when the inhibitor binds to the enzyme, what it will do, it will convert the shape of the active site. Okay, it will convert the shape of the active site such a way that the substrate cannot bind. It will not bind. Or sometimes it will close the structure only. It will close the structure. The active site is completely closed. It will modify the active site. What this will do, the allosteric modulators or regulators, allosteric regulators or modulators, they will change the active site structure. They will modify the active structure such a way that either enzyme will bind with the substrate or it will not bind with the substrate. Okay, either they bind or they will not bind. If it helps in the binding, they are activators. If they inhibit the binding, then it is a inhibitor or inactivator. Okay, sometimes what happens? The binding of this will increase the chance of substrate binding, that is positive allosteric effect. Okay, positive allosteric effect. If the binding of the substrate will decrease the affinity for other substrates, they're called as negative. Okay, negative allosteric effects. Like for example, binding of 2,3-BPG. You might have learnt in physiology, the oxygen dissociation curve. Binding of 2,3-BPG to the hemoglobin will decrease the affinity for oxygen. Yes, that is negative allosteric regulation. A third molecule is binding with the enzyme, because of which the protein is not binding to its substrate, oxygen. Same way in the positive also. When one oxygen molecule binds with the iron, the iron of the hemoglobin, correct? Okay. When the first molecule binds, it will increase the affinity for the second molecule of oxygen. Then further, it will increase the affinity for the third molecule of the oxygen. So there is a positive cooperativity. Okay, there is the increased affinity for this substrate because the first substrate binded. When some molecules, their binding to the substrate will decrease the affinity for the substrate. So 2,3-BPG decreasing the affinity of hemoglobin towards the oxygen, where that will help in the removal of oxygen at the tissue level. At the tissue, you want to release the oxygen for the survival. So the tissue will deliver the oxygen at the site. So this is about the allosteric regulation. There are the examples. Okay, you have to write at least 2 examples in the exam. So what are the activators and inhibitors of some reactions? Like for example, glycogen synthase. It is activated. Okay, it is activated in the presence of glucose 6-phosphate. When glucose 6-phosphate concentration increases, glycogen synthesis starts. That is the activator. One more, for example, acetyl-CoA carboxylase. Okay, acetyl-CoA carboxylase. Who will activate it? Activator is citrate. In the presence of citrate, the enzyme activity increases. It is inhibited by palmityl-CoA, okay, or palmitoic acid. Okay, palmityl-CoA will decrease the rate of reaction, or it acts as the inhibitor for the reaction. Similarly, like for example, others includes say about aspartate. Okay, aspartate transcarbamylase. It is activated by presence of ATP. When there is a lot of ATP, inhibited by the presence of CTP. Okay, so these are the few examples, and also like for example the reactions of your glycolytic pathway. Okay, phosphofructokinase 1, in the presence of lot of fructose 6-phosphate. Okay, or fructose 2,6-bisphosphate. It is activated, and in the presence of the lot of ATP, already energy is high. We don't want to break down the glycolytic process to happen. So it will act as a negative allosteric. Okay, it will decrease the rate of reaction. So this is about the allosteric regulation. So allosteric regulation is happening in the presence of allosteric modulator. What is an allosteric? It is a molecule which will bind to a specific site on the enzyme other than the active site, which is called as allosteric site, and it will regulate the enzyme reaction. Either it will increase the rate of reaction or it will decrease the rate of reaction. So that is about it. Allosteric regulation, which is a 5-mark question. Please remember. The next second type, covalent modification. Okay, covalent modification, it is most commonly irreversible. Okay, it is irreversible change in the structure of the enzyme. So, like for example, we have learned in enzyme something called as zymogens. What are zymogens? They are the inactive form of the active enzymes. Okay, they are the inactive form of the active enzymes. So once you make the changes in the structure, once you make proteolytic conversion of zymogen, that is inactive form of enzyme, into active form of enzyme, you cannot take it back. Okay, you cannot convert the active into inactive and again reuse it. It will be completely removed from the structure. That is denaturation or breakdown of the protein itself. So active enzyme. So this covalent modification is seen with proteolytic enzymes. Okay, by the hydrolysis, these are converted into active form. Like for example, okay, we have most of the enzymes like trypsinogen is converted into trypsin. Okay, trypsinogen is converted into trypsin. Trypsinogen is a zymogen in active form of the active enzyme trypsin. Similarly, pepsinogen converted to pepsin. Fibrinogen, okay, converted to fibrin. So all these are examples for the covalent modification. When you make the hydrolytic changes, that is the hydrolysis of these enzymes, the zymogens, they are converted into the active form of the enzymes. Okay, active form of the enzyme. This is about the irreversible. There are some covalent modifications which can be reversible. Okay, there are some modifications which can be reversible. Like for example, addition of phosphate group. We call it as phosphorylation. Okay, phosphorylation. Or removal of phosphate. That is same, dephosphorylation. Adding of phosphate group and removal of phosphate group to the enzyme will either activate the enzyme or inactivate the enzyme. So that is the reversible type. Like for example, the addition of phosphate group. This is the enzyme. Phosphate is added. It will form a complex phosphorylated enzyme. Phosphorylated enzyme. This is by a set of enzymes called as kinases. Kinases will bring about the addition of phosphate group to the enzyme. Either it will inactivate or activate the enzyme. Similarly, if enzyme is already bound with the phosphate and we are removing it, enzyme plus phosphate separate, dephosphorylation. The common enzyme class who will do this kind of reaction is phosphatase. Okay, phosphatase is the enzyme who will remove the phosphate from the enzyme so that either the enzyme is activated or inactivated. Similarly here also, either it is activated or inactivated or inhibited. So this is about the covalent modification. So covalent modification can be of two types, reversible or irreversible. Okay, reversible modification or the irreversible modification. So this is about the covalent modification. The next type is feedback. Feedback regulation. The feedback regulation plays a very important role in the most of the mechanisms where it is series of reactions. Okay, from A to B, B to C, C to D, and last is the end product. So in such kind of reactions, this plays an important role. The feedback inhibition or feedback regulation plays an important role. Like for example, you are okay, now this is almost end of the summer and you have kept your AC at a temperature of 20 degrees and you're sleeping. Initially, you were good. You are feeling hot because you have come from out. And 20 degrees after 3 hours, you started feeling chilled. You are not able to sleep because it is too cold around you. What you do, you will give the feedback to the AC and increase the temperature to 26 degrees Celsius. So what you did, you regulated the reaction of the AC where it was maintaining the temperature of room of 20, you made it to 26. You have decreased its rate of reaction. You decrease the rate of reaction of the AC. What you did, you are giving feedback. What feedback? That you are already not required this cold temperature around you. You want to reduce it. You want to reduce the temperature, make it to little higher side. So that is the feedback regulation. Okay. So similarly, body also does the same thing. Like for example, A is converted to B, B is converted to C, and C is converted to D. When there is a lot of D synthesized, what it will do? It will give feedback to the first reaction, most commonly the initial reaction, starting reaction. It will tell the enzyme 1, please decrease the rate of reaction because already D is accumulating in the body. So end product is inhibiting the first step in the series of reactions. That is most commonly seen in the feedback regulation. Okay, feedback regulation. Most commonly, if they are inhibiting, then it's called as feedback inhibition, feedback inhibition. If it is inhibiting, it's called as feedback inhibition. So that is about the feedback inhibition. So we have seen the allosteric also, and this is about the feedback inhibition. Now next is the control enzyme synthesis only. How you control the enzyme synthesis, which is long term. Till now, what we have seen is all short term regulations. This is long term regulation. Long term regulation. Sorry, long term regulation. Either you increase the reaction, that is induction, increase the synthesis of enzyme, inducing the enzyme synthesis, or you are repressing. Repressing means you are going to suppress it. Repression. Okay. What is this induction? Induction means it represents. Now we have seen in our very basics of molecular biology that they follow one gene structure. It will have one promoter area. If you activate this, it will increase the synthesis of enzyme or protein. So this is always capped. This is always at the controlled, that is repressed. Always it is repressed. When you de-repress it, when you remove the area and you allow it to react, inducing it. So inducing increases the protein synthesis. Induction increases the protein synthesis. What it will do? It will remove the repressor. That is called as de-repression. Okay, de-repression. Removal of the repressing substance. Normally, this is containing one, say about like cap-like structure, which is closing the site. What we want to do, you want to remove it. You want to de-repress. You want to remove the structure so that the synthesis of the enzyme starts, induction of the enzyme starts. Okay, induction of the enzyme starts. Like for example, you have taken glucose, that is through your diet, through food you have taken a lot of glucose. What insulin will do? Insulin will tell the body, please synthesize lot of enzyme required for the glucose metabolism. Okay, lot of glucokinase enzymes are increased. And also you want to store the glucose. So glycogen synthase or synthetase enzyme is increased. Phosphofructokinase enzymes are increased. So what the insulin is doing is it is increasing the concentration of the enzyme by de-repressing the genome. The gene is de-repressed, enzymes concentration increases, and they start acting on the substrate. So that is about the induction. Similarly, repression. If the synthesis is continuously active, you imagine this is the genome, this is the operator or inducing, and the enzyme is being synthesized. We want to stop it. So we want something to go and repress it. We want something to go and stop the gene expression. So repression also at the gene where it will inhibit. Okay, it will inhibit the synthesis of protein, which is nothing but the enzyme. It will inhibit the synthesis of protein, that is the enzyme. That is like, for example, heme synthesis is one of the most commonly used example. Once heme is synthesized, it will decrease its own synthesis by decreasing the concentration of the ALA synthase. So it will decrease its own synthesis. It's like similar to feedback inhibition, where substrate converting into product is inhibited by the product itself. The product will tell, I am already in sufficient concentration, stop synthesis of me. Okay, if it is required, we'll do, otherwise not required. So that is about the regulation. Okay, how they are regulated long term. Induction and repression by changing the concentration of the enzyme. They will change the concentration of the enzyme. Now, last one is the compartmentalization. Okay, compartmentalization. As I already mentioned, the name itself tells that compartments are there in the cell. What are the compartments? Like for example, cytoplasm is one compartment. Okay, the mitochondria is another compartment. Golgi apparatus one compartment. Endoplasmic reticulum another compartment. Lysosome. Okay, peroxisomes or nucleus. Okay. All these are different compartments, and enzymes are present in these compartments separately. Like what I could tell is, for example, normally we have two types of reaction in the body, metabolic pathways. One is anabolic, other is catabolic. One type of reactions are anabolic, other type of reactions are catabolic reactions. Now, for example, if we take catabolic reaction, beta oxidation. Beta oxidation is nothing but breaking down of the long chain fatty acids or fatty acids in the body for energy purpose. What is the location of beta oxidation? Mitochondria. Okay, beta oxidation occurs in the mitochondria because all the enzymes which are required for the beta oxidation are present in the mitochondria. Now, next, anabolic of same fatty acids, fatty acid synthesis. Okay, fatty acid synthesis. One side breaking down is beta oxidation. Synthesis of same fatty acid is fatty acid synthesis complex. Where is this complex present? Cytoplasm. So you can see the breakdown is happening in one compartment in a different room, and synthesis is happening in another room, cytoplasm, where all the enzymes required for the fatty acid synthesis are present in the cytoplasm. So this is the compartmentalization. You can see that some enzymes are present in one system or one matrix or one environment, cytoplasm. The breakdown, the opposite type of reaction are present in the another compartment. Like if you have beta oxidation and fatty acid synthesis, both in mitochondria. What happens? When you synthesize fatty acid, it is broken into beta oxidation. End products of beta oxidation are utilized for fatty acid synthesis. It becomes a loop that you are wasting the energy. You are wasting the energy. You are wasting the whole reaction pathways. So to reduce that, what body has chosen is they have done a compartmentalization. The particular set of enzymes are present in cytoplasm, like beta oxidation mitochondria, okay, beta oxidation mitochondria, TCA cycle mitochondria, ketone body synthesis mitochondria. Okay, then you have got the electron transport chain mitochondria. Remaining things like, for example, you have got the nucleus. All the reactions related to DNA, RNA, translation, transcription, all are in the nucleus. They don't happen in the cytoplasm. When you see cytoplasm, glycolysis, cytoplasm, okay, then your fatty acid synthesis, cytoplasm. So all these becomes in the pathway. So they are compartmentalized so that it will increase the overall efficiency. Okay, overall efficiency of the metabolic pathways. To maintain that, the body system has made it into different compartments. That is the catabolic and anabolic are occurring at most commonly at the different place. One most commonly used example is fatty acid synthesis and beta oxidation. Beta oxidation occurs in mitochondria. Synthesis of the fatty acids occurs in the cytoplasm, and they are separated by the membrane. So compartmentalized into two different compartments. Okay, so this is about the different types of regulation. So to summarize, the different types of regulation includes the allosteric regulation. Okay, allosteric regulation. Then you have got the covalent modification or covalent regulation. Then you have got the feedback regulation. You have got the induction. Then you have got the repression. And last, you have got the compartmentalization. Okay, so these are the different types of enzyme regulation. I would like to tell you once again, this is an important 5-mark question. This can be a 3-mark question, or this can be a 15-mark question. Please remember. Okay, it can be anything for the exam purpose. Also, please remember, enzyme regulation is different from, okay, enzyme regulation is different from factors affecting enzyme activity. please remember, the both are two different concepts. Okay, both are two different concepts. Enzyme regulation are these 6 regulations. Factors affecting enzyme activity is completely different, where you are going to learn about how the temperature affects, how the pH affects, okay, what does the substrate does on the enzyme, what enzyme concentration does on the rate of reaction, what the inhibitor will do. Okay, you have got competitive inhibitor, non-competitive, reversible, irreversible competition. So all those are factors affecting the enzyme activity. And what we have learned today is the enzyme regulation, how the enzymes are regulated. Please don't get confused in the exams, because whenever I see in the exam answer sheets, students get confused and they write cross answers. Okay, question will be, what are the enzyme regulation pathways? They'll write factors affecting enzyme activity. So they both are completely different and you will be losing the marks. Okay, so please pay attention on what you are writing for the answer. Okay, so this is about the regulation of enzyme activity. So to summarize with the university question, most commonly what they'll ask, briefly explain various mechanisms of the enzyme regulation. So you have tried what is enzyme regulation, what is its significance, and what are the different types of enzyme regulation, and you have to give the example. Same way, they might ask you, write note on allosteric regulation. This can be a 5-mark question in the exam. So allosteric regulation is nothing but the regulation using another site, the allosteric site. So what is allosteric site? It can be a 3-mark question again, allosteric site. So allosteric site is the site other than the active site to which allosteric regulator binds. Either this regulator can be an inhibitor or it can be a activator. Okay, it will increase the rate of reaction or it will decrease the rate of reaction. So that is about the allosteric regulation. Then you have to write the example of the allosteric regulation. Then you have to write a note on compartmentalization and its significance. This is most commonly they'll ask you for 3-mark question. What is compartmentalization? We have seen just now the anabolic enzymes and catabolic enzymes. Most commonly they're present in 2 different compartments. When we tell compartments, the compartments are the cytoplasm, mitochondria, nucleus, all these which are separated by the membrane. They all become one compartment. So compartmentalization. Then sometimes they'll ask you to write a 3-mark question, classify enzymes regulation, or how the enzymes are regulated classification. So just we have seen 2 classes, short-term regulation, long-term regulation. Short-term regulation, you can have the covalent modifications or the non-covalent modifications, which includes even the allosteric regulation. Then long-term, it can be with the help of the hormones, hormonal regulation, or it can be at the genetic level. Okay, that is the induction repression. So at the gene level. So these can be the answer for the questions. Okay, so this is how the most commonly they'll ask on the topics what we have covered today. So with this, we'll be concluding for today's session. That is the enzyme regulation.